Hello and welcome to this webinar. My name is Mayo Meunier and I am a Senior Application Scientist here at Elegen. Thank you all for joining us to learn more about how Elegen's innovative cell-free DNA synthesis technology addresses critical bottlenecks to help accelerate the development of cell and gene therapies. Early 2023, Elegen launched ENFINIA DNA, which allows our customers to order double-stranded linear DNA up to 7 kb. The DNA is NGS verified with over 99.99% per base sequence accuracy. It can be delivered in as fast as 7 business days. Throughout this last year, the team at Elegen has worked to expand the complexity offerings of ENFINIA DNA. Even though at launch, our product was already offering complexity specs that were competitive in the DNA synthesis market, we knew that many applications like cell and gene therapy demanded more. We heard stories from customers about projects dragging on because sequences were overall too complex or because they contained a region particularly challenging to synthesize. We also heard stories of projects where sets of sequences were simply not evaluated because of their complexity. So we launched a limited early access program and began advancing our technology to address those limitations. We worked with key partners to optimize our synthesis process to unlock a new range of complex sequences, and will offer expanded complexity DNA from our e-commerce portal this quarter. The webinar today will dive deeper into our technology and the advancements we've made for this launch. To learn more and receive a notification of the launch, you can visit elegenbio.com/city. Traditionally, making double-stranded synthetic DNA starts with oligosynthesis of small single-stranded pieces, which are usually smaller than 300 bases. Whether chemically or enzymatically made, those oligos have synthesis errors that are carried forward once assembled into longer fragments and eventually into full-length molecules or genes. In order to isolate a perfect molecule, the industry has had to go through cells for the past few decades. Cell-based cloning separates and propagates DNA in plasmid form using bacteria. Once plated, each bacterial colony contains a unique gene. Those candidates are individually picked and prepped to isolate the DNA, which is then sequenced. Screening needs to be repeated until an error-free molecule is found. Many of us have wasted time and money screening dozens of candidates unsuccessfully. Furthermore, cells have the tendency to sometimes generate further errors, particularly in those more complex and difficult to replicate sequences. And finally, some sequences simply cannot be recovered due to cell toxicity, which can be pretty difficult to predict. And this is where Elegen comes in. Elegen has developed a number of novel technologies that allow us to offer some of the longest and most accurate linear double-stranded DNA on the market without the use of cells. With our technology, we have eliminated the need for bacterial-based cloning to produce NGS-verified long and complex DNA in days. Removing the cell-based cloning step reduces factors such as latency, operational complexity, screening costs, contamination risk, potential toxicity, and variability. With ENFINIA, our customers are finding a solution to critical bottlenecks in DNA production and are moving from design to results faster. The main strengths of our ENFINIA DNA product are summarized here with 3 sevens. We can provide DNA as long as 7 kb. Our DNA is built with a superior accuracy with a 1 in 70,000 error rate, NGS verified. And our DNA is built and shipped in as fast as 7 business days. This combination of features brings us at Elegen in the spotlight of the DNA synthesis field by ensuring the highest accuracy for linear DNA on the market without compromising on length or speed of delivery. As you can see here on this graph, other suppliers are not able to produce molecules longer than 3 kb and need at least 10 days to do so. We can synthesize molecules over twice as long as our competitors in the same or less amount of time, and with an error rate over 5 to 30 times lower. Our DNA is received ready to use, as there is no need for a cloning step, either for stitching your full construct together nor to purify your sample before proceeding to your downstream experiments. In just a little over a year since our launch, we successfully synthesized thousands of DNA molecules and proven ourselves for a broad variety of customers spanning diverse applications of the SynBio market. Each of the applications represented here has specific sequences needs, which are bound to various complexity types we are specifically working to address. I'll take the example of the cell and gene therapy space, where Infinia DNA can be leveraged in more than a few ways. With our unique mix of length, speed, and high complexity, customers are able to screen through multiple gene variants, collection of promoters, or other regulatory elements in one go. We've heard customer feedback saying that our technology allowed them to reduce their process time from months to weeks. We have gotten a lot of requests for AAV transferplasmic constructs. Our expanded complexity further strengthens our synthesis capabilities by allowing us to offer commonly used AAV-related plasmid parts such as ITRs and SB40 promoters. Those high GC motifs were previously primarily exploited by customers through purification from existing plasmid, as their synthesis is extremely slow, if possible at all. This quarter, we will begin offering expanded complexity DNA synthesis to all of our customers. The launch increases the ranges of complexity criteria that we offer for Infinia DNA to deliver sequences containing higher and lower GC content, longer repeat regions, hairpins, homopolymers, STRs, among other complexities that are notoriously challenging to make. My colleague Ben will in a little bit dive deeper into how we've improved our processes with examples of the more challenging sequences we have synthesized using our new process. Here, the table on the left shows the details of how we have improved our capabilities in every category of complexity in the span of a year. On the pie chart on the right, we looked at over 11,000 unique sequences that were submitted by customer on our elegenbio.com portal and ranked their analysis complexity scores. Originally, 51% of the sequences did not meet the limits for what we define as standard complexity. Out of these, 30% were classified as high complexity, and 21% were beyond the limits of our original synthesis capabilities. With the new limits, we recover over 60% of the sequences that would have previously been rejected, or 13% of all 11,000 sequences submitted. So if you submitted sequences on our portal in the last year and those were rejected due to complexity, give us another shot, and it is now possible that we can make those happen for you. I've had the pleasure of running our limited early access program for expanded complexity over the past few months, where my team worked with a small subset of our customers representative for a variety of applications. With over 125 unique complex sequences built and counting, we have used our new process to synthesize sequences with notoriously challenging motifs common to SynBio applications listed here, including whole backbone sequences. High GC sequences is what we get the most requests for, and we've made sequences containing regions with upwards of 85% GC for every applications segment. On the other hand, we were also able to build sequences with GC windows of less than 20%, a feature particularly attractive for plants and pathway engineers. For cell and gene therapy, we have produced a variety of ITRs and LTRs for our early access partners, GC-rich but are not offered by any of our competitors, as well as other regulatory elements containing long repeat regions and homopolymers. Those homopolymers and long repeats are generally popular requests across all applications, as these are the most difficult to synthesize without deletions and are also often difficult to sequence as well. In addition to our expanded complexity, we are also introducing a new pricing structure. In the past, we used a fully tiered pricing model of 25 cents per base pair for standard complexity and 35 cents per base pair for high complexity. In other words, if any region within a sequence contained a complexity area beyond the limits for what we consider standard, the price per base of the entire sequence was increased. With the launch, We will no longer increase the price for regions of sequence that are classified as standard complexity if high complexity elements are also present. This dynamic pricing structure means that each sequence will be broken down into regions or standard or high complexity, and only the high complexity region will incur a premium. Our goal here is to provide competitive pricing for both the long and complex DNA, as well as the shorter standard DNA that you may need. With that, just a final reminder of our Infinia DNA specs and of our upcoming expanded complexity DNA launch this quarter. Visit our website to learn more at elegenbio.com/high complexity. Thank you for your time, and I hope to get the chance to collaborate and help you with your project in the near future. I turn it over to Ben for further detailing of our expanded complexity process.
Hello, I'm Ben Allred. I'm a scientist at Elegen, and for the last several months, I have been working with a team that is focused on expanding the sequence complexity that is available in Infinia DNA. I'm looking forward to telling you more about the sequence complexities that are available, some insights into the technical developments that enabled the expansion, and tell you how we are testing the reliability and reproducibility of the product. We are now in the final stages of product development and customer testing, and we're confident that we will release the expanded complexities in early March. The Infinia DNA product begins at our ordering website, goes through the production process and QC, and then is shipped to the customer. The enhanced complexity that I will be presenting represents an expansion of the Infinia product, with the changes integrated into the Infinia ordering website and production process. At the ordering website, sequences pass through many analyses, including identifying the optimal synthesis design. In the past, sequences were rejected that could not be synthesized by our standard complexity or limited high complexity process. The focus of our development over the past several months was to expand the limits of what we could synthesize under our high complexity process and significantly reduce the number of sequences rejected, enabling our customers to order a variety of sequences they could not obtain from other suppliers or easily make themselves. When you look deeper into why complex sequences are so difficult to build, the root cause is primarily inconsistent hybridization of single-strand DNA in assembly reactions. As Marion described in her talk, the starting material for all double-stranded DNA synthesis includes single-strand oligos that are shorter than the target product sequence. The oligos are typically linked together by a process known as polymerase chain assembly, where melting and annealing steps are intended to hybridize complementary portions of oligos in a pool so that they self-assemble into the design sequence. Self-assembly with complexities can fail in many ways. Some examples for the major classes of complexities are shown here. Sequences that have low GC content can fail to hybridize. Sequences with high GC content may hybridize at an alternative location. Long repeats are also challenging since the repetitive sequence typically spans a full oligo, and a single oligo within the repeat is complementary to more than one other oligo. Hairpins or ITRs are another challenge. The individual oligo forming a hairpin may fold onto itself before hybridization, preventing hybridization altogether. To improve our success at synthesizing very complex sequences, we moved away from polymerase chain assembly and developed a novel method for synthesis that controls the exposure of single-stranded DNA during oligo hybridization. Complexities hybridize primarily with their complementary oligos, giving the best chance of correct hybridization. Mishybridization is minimized during self-assembly. We tested this new assembly method that does not rely on polymerase chain assembly or PCA by synthesizing 98 fragments, each less than 1 kilobase in length, representing a wide range of complex sequences. The complexities included repeats at different locations within the sequence, low and high local GC content, and high variations in GC content, hairpins of different length, homopolymers and STRs, or short tandem repeats, and complex sequences that are common to plasmids, as well as sequences that had been previously rejected by our earlier Infinia acceptance criteria. Many of these sequences that we tested were rejected by industry-leading suppliers. We successfully synthesized 92% of these complex sequences, whereas the best performing supplier would only accept 58% of them for synthesis. After observing the potential of the new method, we incorporated this new process into our Infinia DNA production process to test the synthesis of sequences up to 7,000 base pairs long, containing one or more of the complexities in this chart. For the testing, we assembled a collection of 300 complex sequences provided by our customers or pulled from published libraries, and sequences that we designed to systematically cover diverse types and ranges of complexity. In our first phase of testing, we validated the feasibility of expanding our complexity limits by synthesizing all 300 sequences in a manual workflow. We then moved the workflow to our commercial production platform and verified success of adapting the manual method to automation. We selected 120 of the 300 sequences from the feasibility to represent the range and limits of the complexities and synthesis design. After the verification testing, we will conduct a month-long validation process where we are repeatedly producing over 200 sequences that represent well the range of complexities and the range of sequences that customers have submitted to our portal and early access program. We will continue this repeated testing until the end of the month, at which time we will have quantified and validated the reliability and reproducibility. The data is what will inform our confidence that we can deliver expanded complexity at the specifications and time expected by our customers. The results of our verification testing mimicked our early testing and demonstrated that with our improvements, we could synthesize long multi-kilobase sequences containing one or more of the complex sequences at the upper end of our new limits. We ordered 52 sequences we successfully synthesized from 2 other suppliers, and the best performing supplier only accepted 69%. Again, this demonstrates that with our improved high complexity process, we can now offer our customers long and complex DNA not available from other leading suppliers. While we and customers often talk about complexities as individual and independent, the functional sequences that customers need are often more complicated. To test these sequence types, we have included many sequences from early access customers and from libraries such as the SnapGene Plasmid database. Here are examples of some uniquely challenging types of complex sequences contained in our test panels that we have successfully synthesized. These sequences have multiple moderate to high difficulty complexities that are combined in a region. The combinations and relative position can be highly organized for functional elements. Regulatory sequences like enhancers and promoters are notoriously challenging. The examples shown here each have an array of repeats and hairpins at random intervals or very regular intervals. On top of the repeats and hairpins, these also have either very low GC, very high GC, or a variation in GC. ITRs are a specialized hairpin incorporated in every viral vector design and can now be synthesized using our improved methods. Following validation of our new method for full-length DNA production, we finalized new acceptance criteria for Infinia high-complexity DNA. The expanded acceptance criteria comes without changing the requirements for yield or sequence accuracy. I want to highlight that in the new expanded acceptance limits, the local GC percent, which we define as the number of G or C bases in any 100 base pair window within the sequence, will expand from the range of 16 to 80% to the new range of 10 to 85%. Also, repeats will be accepted up to 150 base pairs. Currently, the repeat limit of 100 base pairs has additional requirements for the position of the repeat within the sequence. The limitations on the relative position of repeats will be relaxed so that even 150 base pair repeats that are adjacent to each other, often called direct or tandem repeats, will be accepted. Importantly, our algorithmic limitation on local complexities will be relaxed. Currently, a significant fraction of sequences that are within the March 2023 limits are rejected because the combination of complexities within a window of the sequence prevents successful synthesis. With our new high complexity synthesis process and limits, most sequences that have characteristics within the new limits will be accepted, even when combinations of complexities are localized together. This is just to illustrate my last point about combined complexities. Over 84% of the sequences that we rejected in 2023 contain multiple complex sequences that were at the upper range of our synthesis limits, all within 500 base pair region or extending beyond 500 base pairs. With our new process, we can now synthesize regions as long as 1,000 base pairs containing multiple complexities at the upper end of our acceptance limits. For all the sequences we synthesized in our testing, we measured the synthesis performance and quality of the final product using the same QC methods and metrics we use with sequences delivered to customers. Every sequence is sequenced by NGS to give base, accuracy, and purity, and the product is quantified to give yield. The sequencing data is reported here as a color-coded coverage plot. The blue field represents the reads per base that agree with reference. The other colors represent SNPs and indels. The graph on the left is what we want to see for each sequence we make, and the graphs on the right are examples of sequences with errors that failed QC. For this test sequence with a systematic complexity of 100 base pair tandem repeat, all positions across the sequence agree with reference, and any errors observed are within sequencing noise. Our purity quantification has high sensitivity and shows only 3% of non-target DNA in the well. For this sample, over 3 micrograms would be delivered to the customer, greater than the guarantee of 1 microgram. An early access customer antibody expression sequence had a common combination of complexities, high local GC percent in a repetitive sequence. That prevented us from synthesizing the sequence before development of our expanded high complexity process. With the new process, we were able to make more than a microgram of this sequence with high quality. It was sequence perfect and minimal side products. The sequences characterized here are 2 pieces of an AAV vector, each with an ITR. ITRs are challenging to synthesize because they have stable alternative secondary structure that leads to side products in assembly and amplification reactions typical in DNA synthesis. This quantification shows that our product with the ITR sequence has high per base and side product purity and yield. The enhanced complexity offering I have been presenting will be integrated fully into the Infinia product at launch next month. The focus of our development has been to expand the limits of what can be synthesized and significantly reduce the number of rejected sequences, enabling our customers to order a variety of sequences they could not obtain from other suppliers or make themselves. In parallel to expanding the complexity with the new process, we have been quantifying the limits of the new method and integrating those limits into the website sequence analysis. so that we can provide the customers the most precise view of what they can expect to receive in their order. The product QC and yield will maintain the same Infinia standards as standard complexity sequences. We will continue running samples right up until the launch, but as you can see from the data, our new process for high complexity delivers high accuracy, high purity, complex sequences within our 1 to 3 microgram spec reproducibly. To receive notifications for the launch, please visit elegenbio.com/high complexity or connect with our sales team at info@elegenbio.com. Thank you for joining us. I'll hand it back to our moderator.
Thank you so much, Marion and Ben, for your presentation today. We're now going to move on to our Q&A portion of the webinar. So please do keep your questions coming in. We'll try to get to as many of them as we can during today's session. And any we don't get to today, we will answer by email. So the first question here is for Marion. This audience member asks, do you deliver within 7 days for any type of sequence?
Okay, so for our standard complexity DNA, we usually deliver in 6 to 8 business days. Our high complexity DNA usually takes 10 to 15 business days. And again, for both levels of complexity, we offer up to 7 kb, and both of them are sent out NGS verified.
Great, thank you so much for that answer. The second question here is also for you, Marion. It asks, do you offer DNA with the poly A tail?
So we have shown proof of concept for this, but it is not a product that we offer today. We are, however, planning to launch an early access program for this. And if you're interested, I think you should reach out to our sales team through our website, or you can also send an email to info@elegenbio.com.
Great, thank you so much for providing that info. Our next question here, this one is for you, Ben. This audience member asks, on one of your earlier slides, you mentioned that sequences can contain up to 1,000 BP of complexity. Does that mean you reject any sequence with more than that?
No, the 1,000 base pair limit is specific to localized combination of complexities. If your sequence has a concentration of complex motifs adjacent to each other, spanning a length greater than 1,000 base pairs, it may be rejected. If the complex sequence features are more spread out, the sequence has a better chance of acceptance.
Thank you so much for that answer, Ben. Staying with you here for our next question. This audience member asks, are you using Illumina sequencing for your QC?
Currently, we are using long-read sequencing for our QC. In our development of Infinia DNA, we evaluated a range of sequencing platforms, and for our methods, the long-read sequencing was the best option.
Great, thank you so much for that input, Ben. Staying with you here again for our next question, this audience member asks, can you produce libraries of promoters or other parts for screening?
Yes, we launched an option to order Infinia DNA variant libraries last year. For this, we synthesize a library of sequences that contain 1 or 2 variable regions within an otherwise constant sequence. If the majority of each sequence is constant and you are only varying 1 region, like a promoter, we can build these libraries at a lower cost. Contact us on our website or by email if you're interested.
Great, thank you so much for that. Again, we have another question here for you, Ben. This audience member asks, how long can you make your homopolymers?
We can synthesize A or T homopolymers as long as 30 base pairs and GC homopolymers as long as 15 base pairs. We've been shipping sequences with homopolymers since our launch, and customers have been satisfied. But it's important to understand that currently we cannot guarantee accuracy for stretches above 10 base pairs with our QC methods. So while we can make them, there is some risk with the longer homopolymer stretches.
Great, thank you so much. Marion, our next question here is for you. It asks, do you deliver DNA in a plasmid?
Yeah, so we have been running an early access program to deliver plasmid to select customers. However, we found that many of our mRNA therapeutics customers found that both the length and the complexity of our linear DNA is usually superior to linearized plasmid DNA. And also the fact that the DNA is made through a cellular process is beneficial to their own downstream workflows. However, if you're still interested in plasmid, you can reach out to our sales team through our website, or send an email again to info@elegenbio.com.
Great. Thank you so much for that information, Marion. So Marion, again, we're staying with you for this next question. This audience member has asked a question that sort of has two parts. The first part of the question asks, do you ship to Europe or Asia? And the second part asks, what mass of DNA do you typically ship?
Yes, that's a good question. So while we make all of our product in the US, we have already shipped to several of our customers in Europe, and we are able to ship worldwide. For the second part about the mass of DNA, we do ship 1 to 3 microgram as our standard yield, but we do offer scale-up options, and we are able to synthesize as much as 60 microgram of DNA.
Great. Thank you so much for those answers and giving us that information. Our next question here is for you, Ben. Are there any specific combinations of complexities that are particularly challenging for your technology?
Not really. The complexities we've highlighted, like hairpins and long repeats, challenge all methods used to produce DNA. And we're finding that our technology performs well to the limits that we've published. Aside from my last comment about not being able to QC long homopolymers, our technology is comparable, if not better, at synthesizing complex sequences.
Thank you, Ben, for answering that question and for giving us that insight. And thank you to both Marion and Ben for answering those questions. Unfortunately, that is all we've got time for today. Any questions we didn't get to, we'll be sure to reply to by email. Just a reminder that the webinar will also be available on demand from tomorrow. So please do look out for an email from us with that link. And of course, all that's left to do is to thank Marion and Ben for a great presentation. And thank you to you, the audience, for listening. We hope you'll join us again soon.